We talked about, or one of the characteristics of proteins hormones were that they could be made in advance. Okay? And how you make a hormone is by protein hormone is by gene expression, right? Remember DNA, messenger RNA, right? Atoms in the nucleus, transcription, translation out in the cytoplasm on ribosome, and we get a polypeptide. Now, I'm not going to call it a protein because you know to be a protein, a polypeptide has to fold, primary structure, secondary structure, tertiary structure, get some three-dimensional shape, have active sites or whatever. Right? Then a polypeptide, right, we'll just say this, has to be modified for use. If it's a protein, we fold it to 3 dimensions. If it's a hormone, we may cut pieces of it up and to make it active, right? So this modification doesn't have to happen at the same time as gene expression. Meaning you can do gene expression and store something in an inactive polypeptide form, right? And then only do the modifications when you need it. Okay? So this is what we call post-translational modification, right? Post, after translational, translation. And you modify. Now this first thing that gets produced right here, if we're ultimately going to modify this and produce a hormone, here. This first thing that would get produced right here would be called a prohormone. Pro means before. Okay. And then the prohormone gets modified into the hormone. What's the modification? Usually involves, you start with a long polypeptide chain like this. You might have to clip a piece of it out like that. And then you might have to take this piece right here, stick it on that. It just depends. Right? That's actually what happened with this one. But it depends on what we're looking at for that. So the body would make a prohormone and then modify it into the functional hormone. Right? So this is functional. Sometimes, this becomes actually pretty silly if you ask me, because it's just terminology. Sometimes there's 2 modification steps that have to occur. So there is actually something that occurs before the prohormone. And again, like I said, I think this is silly. So we would call it the pre-prohormone. Again, it's just a name, but it's more than the name. What's important to understand is just simply that, and this is logical, when you first make something from the process of gene expression, it's probably not functional. So that modification can be, take the polypeptide, the prohormone, modify it, now you've got the hormone. Or that could be, take the polypeptide, modify it, it's still inactive, store it. That's the pre-prohormone, now we made it a prohormone. And then when you want to do it, activate the prohormone. It doesn't really matter, right? It's just a multi-step process. So we're just trying to get to the point where we can have this thing active. And what makes it active is simply this, right? Is that it can bind to the surface membrane receptor. Because remember, don't forget, this is a protein-ligand interaction, right? So we've got specificity, we have affinity. That's the thing we're getting at right here, is we have affinity. What makes the hormone active is that it is at a high affinity to the receptor. And it will then bind to the receptor and trigger the response. Okay? And often through this signal transduction, which just means A happens, then B happens, and C happens, and whatever. That's all we're looking at.